Hello, everybody, and welcome to the virtual seminar series on Gaussian processes, spatiotemporal modeling, and decision-making systems. And we're very excited to have everybody here, and thank you very much for joining us. My name is Alex Turunen. I'm a postdoc at the University of Cambridge, and I'm going to be one of the organizers that's hosting today. And I'd like to thank my fellow organizers, Jeff Pleiss, Lisa Simonova, and Xi Wang for helping put the series together. And so just before we get started, I'm going to make a few announcements. The first off is that there's one more talk coming up to sort of conclude the seminar series, if I'm remembering that right. And you can check that out on our website, which is gp-seminar-series.github.io. And you can sign up there to receive email links to join these seminars via Zoom. And so, feel free to do that if you're not registered yet. Or you could also join via YouTube, which does not require any registration. So that's just available for anybody who's interested in seeing the seminars. And that includes after they've been recorded. So if you're kind of not joining us live, then you can instead see them later. Seminars take place at 1600 UTC, which you can check what that is in your local time zone. If you're joining via Zoom, you can use the raise your hand option to ask questions, or you can type your questions into the chat. And the speaker can either choose to kind of pause and take questions, or they can choose to hold them at the end, sort of depending on their preferences. And with all of these organizational announcements said, it's my great pleasure to introduce Diana Kai, who is at Princeton University and will give today's seminar. Diana, the floor is yours.
All right. Yeah, so thanks so much for the introduction and for inviting me to speak in the seminar today. So I'll be talking about multi-fidelity scientific discovery and design, and in particular, the methods for doing discovery and design. A lot of scientific applications are expensive, but in a lot of cases, we actually have access to different fidelities of models and measurements that trade off cost and accuracy. And so I'll talk about how we might use some of these in methods for doing experimental design and also Markov chain Monte Carlo. And so this is joint work with collaborators, Andy Jones, Didong Lee, Barbara Engelhard, and Ryan Adams. So we're seeing this. We're seeing a lot of applications for scientific discovery and design. They kind of increase of development in machine learning and computational methods. And so some of the domains that I am interested in, and we'll talk briefly about today, include trying to design spatial genomics experiments, and also designing kind of materials such as energy materials. And finally, there's a whole class of problems in the physical sciences that involve doing sort of inverse parameter estimation in design, such as trying to understand cosmological parameters to trying to design structural materials. And so there's some common challenges that we often face. And one is that doing experiments in a lot of these domains is expensive. And so, as I mentioned before, we're increasingly turning more and more to the use of computational methods. But even in those cases, we kind of face a lot of challenges in terms of dealing with really expensive simulations, complex modeling assumptions, and expensive numerical approximations. And so, as I mentioned before, we're sort of also being able to make decisions and trade-offs about what sort of fidelity to observe different kinds of observations and simulations. So just to give some examples, in the genomic setting, there's a lot of different kinds of sequencing technologies available and different types of resolutions, and they all trade off various kinds of costs. They sort of trade off kind of costs with the accuracy of the actual information that you get out of it. In the sort of material science setting, we are often dealing with computational methods and simulations, although we also kind of observe different fidelities of observations from, say, computation experiments. But in terms of just thinking about simulation alone, we can also think about simulating different properties at different fidelities that are perhaps cheaper and less accurate versus more accurate, but more expensive. And so these types of simulations occur in various other kinds of applications, such as trying to simulate properties of structural materials to cosmological phenomena. So today what I'll talk about is thinking of this question of how can we more efficiently select which experiments to run. In particular, I'm going to focus on this application of doing spatial genomics experiments. And here, what we're going to do is we're going to talk about a multi-fidelity active experimental design framework that uses tempered likelihoods. And I'll also talk about how we can develop general MCMC algorithms using multi-fidelity models. And I'll show how we can leverage a spectrum of increasingly complex models to get exact estimates. So to give a roadmap of the talk, I'll first talk about this experimental design setting and motivate and formalize designing spatial genomics experiments. Then I'll talk about our actual multi-fidelity experimental design framework and give some preliminary results. And finally, I'll give a really brief primer into some related applications in active search. And then the second part, I'll talk about doing parameter estimation design using Markov chain Monte Carlo type methods. And how we can actually utilize this sort of sequence of low fidelity models by considering a randomized fidelity model to get sort of exact estimates. And finally, I'll talk about some applications to various domains, including some Gaussian process modeling setups. All right. So to motivate the sort of experimental design problem, I'm thinking about the sort of spatial genomics problem. And so this is sort of a relatively new sequencing technology where you can now sequence the gene expression of some tissue at various different spatial locations. And so there's several applications where you might use this technology. For instance, you might try to construct an atlas of some tissue, for instance, of a brain. And so what this will allow you to do is query some spatial location and then return some detailed description of the phenotype, for instance, different cell types associated with that. And so the idea is this might help with understanding the genetic basis of various diseases, such as human neurological disorders. And so this is sort of an example given by the Allen Brain Atlas, where they sort of constructed this atlas of a mouse brain. A second kind of application is trying to localize and profile prostate tumors, or just sort of some region of interest more generally. So here the problem involves studying a biopsy sample from tumor tissues. And so what we'd like to do is identify the border between the tumor and healthy tissue using as few sort of slices of the tissue as possible. And so this localization problem is a common goal in things like cancer pathology. And so in particular, doing these experiments is really expensive. According to this article, to construct this brain atlas itself costs roughly $41 million. And in general, it's very expensive to do these experiments. And so today, what we're going to think about is, are there more efficient ways of doing these experiments? And can we more efficiently choose which tissue slices to profile. And so the way we'll think about this is through a Bayesian optimal experimental design framework. So the typical sort of spatial genomic sequencing setup looks something like the following, where you have some tissue and you would slice it into a lot of really thin slices. And so here's an example of one of these cross sections that you might get out of this brain. And so then what you would do is you would have, you would sequence this to get gene expression information at these individual spatial locations. And so there's a lot of effort in terms of then trying to come up with various models for this sort of new type of data set. And so our motivating question is, can we more efficiently choose which tissue slices to profile? So the standard thing might be to sort of serially slice across like this, and then perhaps sequentially just sequence these various slices. There's also the sort of issue that there's various kinds of sequencing technologies that we might be able to choose that vary in cost and quality. And so can we also incorporate these into some sort of sequential experimental design framework where we might start by choosing some design, then running some experiment and observing the results. In our case, perhaps that might involve sequencing the gene expression and then updating sort of our model of the problem. And so here, when it comes to actually choosing the design, the kinds of questions we're thinking about are, what slice should we actually take here, and at what fidelity should we kind of do the sequencing of that gene expression? So to think about this question a little bit more, essentially, we're trying to think about what kinds of slices we might take. And so when we take a slice, we're sequencing its gene expression. And what we'd then like to do is, instead of taking all possible slices and sequencing them sequentially, you might imagine then trying to infer the surrounding observations of the unobserved parts of the gene expression using the slices that you've gathered. And the idea here is that there might be various redundancies or correlations between nearby slices that we'd like to take into account. So in our setting, the way we think about this is by having some sort of spatial domain that represents, say, a three-dimensional tissue. And then when we go to actually take a slice, we sort of observe some gene expression. And so one of the simplest ways we might model this is by using a Gaussian process, and then kind of using then actually, when we get these gene expression levels at each of these discrete locations, modeling that as Gaussian with some noise model, and then trying to sort of use that to then impute these unobserved observations. And so we could imagine various kinds of design spaces that characterize, the set of possible slices we might take. For instance, the one I mentioned before is thinking about these parallel slices, and what we're going to call kind of throughout the talk as the serial slicing strategy refers to taking these parallel slices sequentially. But alternatively, we might also try to come up with some sort of utility function that allows us to choose slices. For instance, you might consider this really kind of unconventional slice that looks something like this. And so in order to do so, we have to come up with some kind of design space. So for example, one design space might represent all of the parallel slices that I described earlier. Another kind of design space we might consider are, say, two-dimensional planes in this spatial domain, perhaps parameterized by some, slope and intercept. And so then what we would do is at each experimental iteration, we choose a slice in our design space that maximizes the utility of the slice. We observe some gene expression and update our model. And we're going to first just try for this work. What we did was we tried this sort of simplest type of expected information gain utility, where here we're measuring the expected value of the entropy of our model sort of before and after observing this sort of slice and the associated gene expression. So we tried this on several of the data sets that I mentioned earlier. So the first sort of setting is trying to reconstruct this Allen brain atlas. So here we have the sort of question is, can we reconstruct this atlas with fewer slices? And what we have for our data are a bunch of gene expression at various spatial coordinates. And for the purpose of this experiment, we just sort of looked at a single gene here. So that's what these colors represent, the sort of expression levels of that one gene. And so a hypothetical slice might look something like the following, where once we've taken that slice, this is a visualization of the actual gene expression of that single slice. And so the goal here is, again, to sort of impute these other kind of unobserved regions using the slices that we've taken. And we sort of measured this we measured the actual reconstruction using R². And so here what we are showing on the x-axis is the sort of experimental iteration, and on the y-axis is this reconstruction. And so what we find is that these sort of using the sort of information gain based utility functions versus we're basically able to kind of get better reconstructions than the sort of serial strategy where you're just sort of slicing everything parallel sequentially. The second application is this tumor localization problem I mentioned earlier, where the goal is to try to identify the border between tumor and healthy tissue. And we'd like to sort of do this by collecting, fewer slices than in the serial slicing strategy. So in this picture here, what I'm showing is this is a histology slide, which then has overlaid on top of it the pathologist's annotation of the sort of healthy and cancerous tissues. So the part in blue represents the tumor. And we again have this sort of gene expression data at various spatial locations and kind of repeat that same process from before. But this time we have sort of a slightly different model that I haven't really mentioned, but it's essentially like a generalization of a sort of logistic regression type model. And so using that, we're sort of trying to then classify whether or not it's if there's a tumor there or not. And so based on that approach, we're able to sort of get to better classifications in fewer iterations. And so this is in comparison with this sort of serial slicing strategy that I've been talking about so far. And so this is also sort of an example of under sort of our approach, what we get after sort of 5 iterations of this design. And so this sort of represents the tumor probability, and you can kind of see this region corresponding to the sort of region in the annotation. Okay, so now we might think about how can we incorporate multi-fidelity information into this. So if you recall before, I was sort of saying there are different kinds of sequencing technologies and sequencing resolutions that we might consider actually collecting our gene expression at. And so so far we thought about a very simplified sort of setting where we're just thinking about this fidelity denoting some kind of spatial resolution. And the way we set this up is we have a set of fidelities that are kind of values between 0 and 1 representing our sequencing resolution. And we now augment the design space to be sort of possible slices by fidelity, or in this case, the resolution. And we also kind of consider the actual cost of observing a design at a particular fidelity. And so then what we have sort of at every iteration of our design are some observations that correspond to the slice locations and the gene expression, as well as the sort of fidelities at which we took those slices or sequenced the gene expression. And so we consider a sort of slightly new kind of utility function now that we call the sort of multi-fidelity expected information gain. And the idea is really similar to in the single fidelity case, but here we're sort of now measuring what's called it's sort of like it's a slightly different model that is essentially a multi-fidelity posterior, and I'll say a little bit more about this on the next slide. But it's some sort of notion that measures a multi-fidelity posterior before kind of observing our design versus after observing the design. And the final objective then kind of involves taking this multi-fidelity expected information gain and then dividing by the actual cost of observing the design at a particular fidelity. And so we call this sort of multi-fidelity expected information rate, and that's sort of what we end up using later on. And so what is this multi-fidelity posterior distribution? So this is sort of a pseudo posterior where we're now tempering our likelihoods. And the idea here is that we have some data and we're raising the likelihood to some power. And the interpretation of the power is sort of controlling sort of how concentrated or how much weight we want to give that data point, essentially. And so basically, smaller powers kind of correspond to higher noise levels. And so here's just sort of a plot that demonstrates what this means, where we assume we have like two fidelities, one high fidelity, where we give all the high fidelities a power of 1, and a low fidelity, which we vary. And just to kind of look at one of these plots, you can sort of see in this gray curve, if half of our data are low fidelity and half of our data are, if half of the data are low fidelity and half are high fidelity, then if we vary the power, it will kind of look like the following 3 panels, where the main point I want to illustrate is that as we increase this power, the actual posterior here gets more and more concentrated. And so that's kind of what we're using to model the different noise levels. And so this is sort of very preliminary results, but what we thought about was a simplified 2D spatial genomics application where basically the reason for this is this data set comes in these sort of slices. And so because we can't, simulate alternative slices here, what we do instead is consider, just one-dimensional slices in this two-dimensional cross-section. And so we kind of artificially compute different fidelities based on subsampling different levels of points for each of these slices. And essentially, here's like a very simple version of this where we're able to kind of reconstruct the single fidelity designs in this multi-fidelity framework using these sort of lower fidelity observations. And there's a lot more to do here. In particular, we need to think about more complex design spaces that we did kind of consider in the single fidelity case that accounts for things like tissue fragmenting. For instance, the sort of crossing designs like this are not practical because it would sort of fragment this tissue in kind of implausible manners. So that's something that we're actively working on, and also trying to think about more realistic ways of combining different fidelities of actual sequencing technologies. So very briefly, I want to mention there's sort of a lot of other applications where thinking about these design problems is relevant. So one problem I'm working on is trying to design these energy materials. And so the goal here is to design novel alloys with certain desirable properties. And so here we're usually operating in sort of a computational environment where we're trying to do these simulations that help us simulate properties using quantum chemistry. And so these kinds of simulations are very expensive, and in general, thinking about kind of how can we more efficiently do this is something important. And so currently, I'm working with some material scientists and other computer scientists to develop physics-based active search methods to help discover stable alloys. And here, what we're basically trying to do is augment existing active search methods based on Gaussian processes. And the problem there is the sort of typical thing tends to encourage more exploration based on the sort of posterior uncertainty. And so in this particular application, we end up selecting more unstable alloy compositions. And so the points that are actually that we'd like to select are the ones that are sort of tight with the actual convex hull of this problem. And so what we're developing are these sort of physics-aware active search methods that take into account the structure and underlying physics of this sort of convex hull problem. And so here we're able to then do kind of select more compositions that are actually near the hull. And so, of course, there's a lot of different sort of multi-fidelity simulations that we might actually perform. And so I think later on down the line, we'd really like to think about how to actually incorporate those into our actual search problem. Okay, so as I was saying before, the sort of main problem is that doing these experiments or simulations are very expensive. And so I mostly focused on this application in spatial genomics, and we showed how we can more efficiently design these spatial genomics experiments. In particular, we show that our approach kind of is able to do better on these sorts of atlas building objectives in this tumor localization problem using fewer slices compared to sort of traditional serial slicing strategies. And so there's a lot of different future directions. In particular, the sort of multi-fidelity side of things is just sort of getting started. And so we'd really like to think about incorporating different spatially resolved measurement technologies that are available and sort of really think carefully about how to incorporate their sort of costs and different spatial resolutions and levels of precision. Another sort of avenue is to explore different design strategies and objectives. So here we looked at a very sort of simple model using essentially a Gaussian process regression type model plus expected information gain. And in practice, I think there's a lot of more complex modeling tasks that we might like to perform with, say, models of latent structure. And finally, we'd also like to compare with some more traditional multi-fidelity approaches, such as using multi-output Gaussian processes. And so generally, we expect to have slightly different properties in our setting. And so that's something we're actively working on. Okay. So I just talked about this sort of problem of trying to select experiments. And so in the second part, I'll focus on trying to develop general MCMC algorithms using multi-fidelity models. And I'll show how to leverage a spectrum of increasingly complex models to get exact estimates. So MCMC is a widely used task in a lot of different scientific computing applications. For example, we might like to estimate physical parameters and report their uncertainties. And we could also use it in optimization problems to design materials that have certain particular properties. And so in a lot of cases, we have some, say, simulator of a physical system where the model becomes arbitrarily more accurate as we give it more compute. For instance, you might have some target density that you would like to kind of work with, where this depends on the solution to some differential equation, and it gets more and more accurate as your discretization of the problem gets finer. But at the same time, it also gets more expensive to compute. And so the reality is our models are going to be imperfect due to having limited computation. And so you're going to get biased estimates out of a lot of existing algorithms. Especially, the typical way is you pick some discretization and work with that. And so what I'm going to show is how we can design general MCMC algorithms that automatically navigates the spectrum of coarse to fine-grained approximate models to obtain estimates that are asymptotically exact. So in particular, just to give a really simple example, we might consider this Metropolis-Hastings algorithm where here we're trying to simulate samples from some desired target density pi. And the way you would do this is iterate through the following steps where you propose some state coming from some proposal distribution and then accept or reject that proposed state with some probability. And the main thing I want to point out is that this ratio here depends on the evaluation of our target. Evaluating it repeatedly in this sort of inner loop becomes impractical when this function is very expensive to evaluate. And so more generally, all MCMC algorithms tend to have this flavor where you have to evaluate pi up to some normalization constant. And so, earlier I gave this example where pi might depend on the solution of a differential equation. And so that's sort of very common in a lot of models in science. Here you also might have problems that depend on things such as a function's minimum or some intractable integral. And in all of these cases, you have some model evaluation that has this compute accuracy trade-off. And so in practice, you use some approximate model by evaluating your pi at some fixed fidelity. And so this is going to introduce some bias. However, our models can often be evaluated at a sequence of fidelities where the model becomes more and more accurate, but also more costly. And so sort of at one side of the spectrum is the sort of coarse-grained model versus the fine-grained model. And so you can imagine giving each of these names, pi 1, pi 2, pi 3, et cetera, and consider the sequence of models induced by varying the fidelity. And then kind of consider kind of this pi infinity that represents some perfect fidelity model as a limit of this sequence of low fidelity models. And so the typical thing people do is they'll choose some low fidelity model pi and use this as their target density with MCMC. And so this is going to introduce bias from this discretization error. And it also gets more and more expensive as we refine the fidelity. But we're seeing this growing literature of actual sort of methods that are trying to combine multiple fidelities of models. So these are multi-fidelity MCMC models that are choosing to use, for instance, one expensive and one cheap model. For instance, you might use the cheap model as some low-pass filter before making the more expensive evaluation. And so there's sort of various versions of this, but in general, you need to choose some finite number of fidelities. And so there is going to be some bias introduced here. And also in a lot of the sort of algorithms that people have considered in practice for multi-fidelity models, they're typically specialized to Metropolis-Hastings, and there hasn't been a whole lot of exploration outside of that. And so kind of inspired by these existing multi-fidelity methods, what I'm going to tell you now is how we can design general multi-fidelity MCMC algorithms that, can kind of work for various kinds of MCMC algorithms and not just for MH. But here, it's going to automatically utilize this entire sequence of models to get estimates as though we had access to this perfect fidelity model, pi infinity. So here's what the basic recipe looks like. As the first step, we're going to construct a randomized fidelity estimator that is perfect fidelity in expectation. So here we have some pi hat of k of theta. And here, what k represents is some random fidelity. And it has the property that if we take its expectation with respect to some distribution mu, we're going to get back this perfect fidelity model. And so mu is a distribution on the fidelity k that we get to choose. And I'll say a little bit more about that later on. The second step would then be to use the estimator above, so this pi hat of k, as our target density instead of the perfect fidelity model in our MCMC procedure. And so what we're going to do here is simulate samples from the perfect fidelity model without ever actually having to evaluate it. And so it might be a little bit surprising why this is possible. And so essentially what we're doing is constructing a Markov chain on the following state and fidelity, where here we have some joint target distribution on the fidelity in our state equal to our mu of k, which is this distribution of the fidelity multiplied by our randomized fidelity estimator. And so if you then go and compute the marginal distribution of our state by summing over all possible fidelities k, it looks something like the following, where if we sum over this right-hand side, we note that this is just the expectation of our estimator with respect to mu. And sort of by construction from step one, we are just getting back the infinite fidelity model. And so essentially, by constructing the Markov chain on here, we're then able to get samples from this perfect fidelity model, but we only ever have to evaluate this randomized fidelity estimator that is going to be cheaper in our setting. And so this general technique of replacing this target with some unbiased estimator is called pseudo-marginal MCMC. And so it's been used in like a variety of other contexts. And so here we're showing how to apply it to this multi-fidelity setting. And to actually do this in practice, what we're going to do is, instead of kind of jointly sampling both of these at the same time, we're going to alternate between sampling from the different conditionals of k given theta and theta given k. And so this has some practical benefits in terms of perhaps avoiding sort of the sort of classic sticky behavior that is common in these pseudo-marginal techniques. But it also allows us to basically break it down into an update with this sort of k given theta conditional, but also this theta given k conditional. And in particular, the second one is now sort of treating our random fidelity as this, we're now conditioning on our random fidelity. And so this is essentially like doing the usual deterministic MCMC update. And so we can kind of choose any any of our favorite MCMC algorithms to use here. And so this makes it very general to apply to problems in practice. So now, how do we actually construct this randomized fidelity estimator as mentioned in step one? So the way we're going to do that here is by rewriting the perfect fidelity model, which is this limit of a sequence of low fidelity models, in terms of this telescoping series. So here, we're going to take kind of pi k minus 1. And of course, pi 0 is just 0. And so here, all we've done is taken our perfect fidelity model and rewritten it in terms of this sum. And you can kind of see this by starting from pi 1 and then kind of iteratively going through the terms. So here we add pi 2 and subtract pi 1. And so these two terms cancel. And then we would add pi 3 and minus pi 2, and these two terms cancel. And so you can kind of see how you get back this original sequence. And so to actually get the randomized fidelity estimator, what we're going to do is take a random truncation of this infinite sum. So in particular, we truncated at k and reweight the terms such that it's still going to be unbiased. So here, this WK are some weights that we can choose, such that when we take the expectation of our pi hat, we get back pi infinity. And so one of the simplest ways we might do this is by choosing these weights here. So here, this is just kind of 1 over the probability of k multiplied by an indicator variable. But the most simple explanation of this is just that we are simply subsampling a single term of this infinite sum. And so mu here is like our proposal distribution. And so you can rewrite this by canceling out all the other terms. And you just get this single term here. And so this has been very easy to see that if you were to then take its expectation with respect to mu, you would get back the sort of infinite fidelity model. And so it's unbiased. And so there's a sort of whole class of other estimators you might consider by choosing different weights. And in our experiments, we consider something called a Russian roulette estimator. There is one caveat that I won't get into right now, but you can get negative estimators here. And so there are some additional tricks needed to actually get this to work in practice. And so essentially, we're able to do it in a way such that we're still getting these sort of asymptotically unbiased estimates of the sort of Monte Carlo estimate that we're trying to get. Okay, so that's the basic recipe for how to actually use this estimator, construct the estimator, and then now I'm going to talk about some examples. And previously, I was sort of giving this example with differential equations and varying the fidelity or the discretization of the problem. But there's sort of other kinds of discretizations or other kinds of computations you might also imagine where you might be able to construct such a sequence. So one example is in this log Gaussian Cox process model. So this is a model where you have some rate lambda given by e to the f, where f is some basically is coming from a Gaussian process. And so this is used widely in a bunch of spatial and spatial temporal point process applications. And you have a likelihood that looks something like the following, where it has this intractable interval. And so the typical approach to handling this is some kind of approximation of the interval using a discretization of the space. For instance, you might do some simple binning, or you might use some kind of other quadrature rule to estimate this interval. And so one example we looked at was this kind of classic test problem of trying to estimate the rate in these sort of coal mining disasters data set. So here what we're doing is looking at applying this log Gaussian Cox process model to this data set, where the way we are representing the fidelity is by the number of quadrature points. So what we're going to do is we take some quadrature estimate of that integral and likelihood, and we construct this sort of sequence of kind of increasing fidelity models by increasing this number of quadrature points. And for our MCMC algorithm, we use something called elliptical slice sampling. And so this is an algorithm that is nice if you have these sort of like high dimensional Gaussian, latent Gaussian models. And so essentially, we basically looked at this multi-fidelity elliptical slice sampling algorithm and compared it to some single fidelity elliptical slice sampling problems where you can see that for very low values of k, where k represents the number of quadrature points, that there's some sort of, I guess, some bias that is fairly obvious in this plot as we sort of kind of use more compute. But essentially for the higher values of k and for the multi-fidelity elliptical slice sampling method, they all sort of end up in the same place, but we're able to sort of get there a little bit faster by combining various fidelities of models. And we also considered a bunch of other kinds of applications of multi-fidelity MCMC. And so the first one I talked about was this quadrature example, but I also looked at something like this ODE system parameter identification problem. So essentially, you have some likelihood that depends on a solution to an ODE. And so we'd like to then sort of do Bayesian inference of the actual parameters of that ODE. And so the fidelity here represents the time step discretization of the ODE. We also looked at sort of Gaussian process hyperparameter inference, where the fidelity represents the number of iterations in our conjugate gradient solver. And then another application we looked at was an optimization application where the problem has you have some objective that depends on the solution to a partial differential equation. And so here, you can imagine the fidelity representing the spatial discretization of the problem. So just to wrap up, we have models that are going to be imperfect due to having limited compute. And so this leads to biased answers. And so in a lot of settings, we actually have access to a sequence of models that get arbitrarily more complex with more compute. And so we showed how to design general multi-fidelity MCMC algorithms to get estimates as though we had access to the most complex, expensive model. And so there's a lot of future directions that I'm working on, in particular, trying to apply this to particular classes of problems where we might be able to analyze and also particular application domains, for instance, designing structural materials, but also other kinds of simulation-based inference problems. And yeah, in general, I think there's more work that needs to be done in terms of making this more robust for large-scale problems. And a lot of this has to do with variance reduction. So here are some of the references for the work I talked about. The first two are on the sort of experimental design problem I talked about earlier. And then the third one is about this multi-fidelity MCMC method. And finally, the last one is a paper about change point detection. I didn't talk about it today, but this idea of this tempered multi-fidelity posterior kind of was explored in this paper as well. Okay, yeah, any questions?
And so I wanted to start by saying, thanking Diana for today's talk. And so we're going to just now move on to the question answer period. And the way that that's going to work is there will be like two parts of it. There will be a part that's recorded, and then at some point we'll stop the recording. And for anybody that sort of prefers not to be recording their question, and you can either use the chat or the raise your hand option in Zoom in order to ask your question. And so I guess, do we have anything to start off? You see there's a question by James who I've asked to unmute.
Hi there, can you hear me?
Yes.
Excellent, thank you very much. It was a really interesting talk. I didn't quite understand the notion of this perfect fidelity model. Does that mean that you've effectively computing everywhere within your parameter space? Or is it more of like given that you can only have in practice a fixed fidelity, you can only go up to a certain point which matches your observations or your data that you've seen. So yeah, can you clarify which of the two, or maybe I'm misunderstanding something?
Yeah, so it has to, I guess it's application dependent, but I think the sort of example that I was talking about with this integration problem is sort of the perfect fidelity model is this involves this likelihood where you have the full integral, and then the sequence of low fidelity models there correspond to estimating the likelihood using a quadrature rule that only has a finite number of quadrature points that's increasing in fidelity kind of as we go on in the sequence. And the sort of in this sort of setting here, we're generally thinking about for the differential equation examples, also a discretization of your space and kind of taking that discretization level to be smaller and smaller until we're essentially taking that error to 0. And so that's sort of what the sort of perfect fidelity model is, sort of the limiting model. Does that make sense?
I think so. So if you could only evaluate up to a certain point, you might find that eventually you start getting discretization errors. So your perfect model would then be at some point before you get to your discretization errors.
Yeah, I guess the way I'm thinking about it is like, if we were basically the error is going to get worse and worse as we kind of or I guess the error is getting smaller and smaller as we refine the fidelity. And so we're sort of thinking about the limiting model as that error is going to 0. There are some applications where perhaps you are only dealing with a finite number of, for example, when we're thinking about something like conjugate gradient, we're typically only dealing with a finite number of iterations in terms of when that is converging. But for a very, very large system, we might think about effectively only being able to run a small number of iterations or a much smaller number than the total size of the system.
Yeah, it was excellent. Thank you.
So I guess perhaps we have another question before we conclude the Zoom, the YouTube, excuse me, broadcasting. If anybody else would like to ask something. Okay, so in the absence of this, I'm going to go ahead and make the concluding announcement so that we can conclude the live stream. And before that, I just want to thank all of our listeners on YouTube for joining us and remind everybody that we have another seminar coming up next week. And so you can check out the schedule on our website, which is gp-seminar-series.github.io. And please feel free to take a look at that and join us